probably everyone. It's probably given enough time now. Okay, so good afternoon, everyone. I'm Anamika. I'm your host today. I'm just going to try and... All right, so I am Anamika. I'm a field scientist for Chemometec. Chemometec is based in Denmark, and it's a global manufacturer of cell counting instrumentation. We work closely with our customers and increasingly in the field of cell and gene therapy. We have observed over the last few years some of the typical challenges that some small companies are facing when they try to upscale their process to manufacturing. So with this in mind, we thought we would invite Matthew Smart, Senior Scientist with Cell and Gene Therapy Catapult, and James Kusina, Head of Bioprocessing from Microfluidics, to share their experiences and their insights into upscaling. Just some housekeeping notes before I hand over to Matt first. The presentation, and we'll launch a poll between the presentations, but the presentations and polls should be about 30 to 35 minutes. And then we'll host a Q&A session, which will last about 15 minutes. We would like you to type your questions into the Q&A box, and we will do our best to get through all the questions at the end. But if we run out of time, we will be in touch via email to answer them. So without further ado, I will hand over to Matt, and Matt will do the presentation.
Thanks, Anamika. Thank you for inviting me to talk today. So my name is Matthew Smart. I'm from the Cell and Gene Therapy Catapult. I'm going to give a quick overview of some of the key considerations in developing an ATMP manufacturing process. So just to begin, a quick introduction to the Cell and Gene Therapy Catapult for those of you who haven't heard of us. We're essentially a not-for-profit within the UK, and our remit is to accelerate the commercialization of cell and gene therapies. And we're able to do this by supporting in a number of ways. We have a unique technical expertise and facilities at our disposal, and we can work with the whole array of the cell and gene therapy space in a number of different ways. We do this from our 3 sites. So we have a development centre in London, where our industrialisation team is based. We have a manufacturing facility in Stevenage, where people are starting to manufacture their first batches for market. And then we've recently acquired a Braintree site, which is going to be focusing on tech transfer innovation for industrial scale manufacture. And as I say, we support right across the clinical development pipeline. So everything from industrialization, things like process and analytical development, supply chain, the regulatory side of things, and regulatory, non-clinical, CMC, etc. And then also on the, oh, yeah?
I don't think you're sharing your screen.
Oh, okay. Apologies, everyone.
It's okay. There we go.
There we go. Okay, great. Sorry about that. Okay. So yeah, and then on the engagement side, we're uniquely placed within the industry to form collaborations between various stakeholders. We do health economics and reimbursement and IP, etc. So we can really support you right throughout the process of development. So I'm going to be talking mainly on the technical side of things. So where to start basically in developing your cell and gene therapy manufacturing process. So I think the most important thing, and we have it regularly with collaborators, is can you try and understand as much as possible in that early discovery phase as you're developing your process. The context of how you approach various problems, you might not need to deal with them straight away, but I think it's a really good idea, a really good plan to just start to think about what your product's going to look like, how that process is going to be impacted based on that kind of understanding. And, this is going to be really impacted by your short, medium term and long term goals. So, it can be a living process, but I think it can often have a profound effect on how your process progresses through development. So just as a few examples here, things like, what's your process monitoring look like? Can I get my process to be animal product free, xeno-free early on in that development? Do I have enough, can I make enough product with my current process, etc. So, it's all about putting that plan in place so you'll really start to think okay, what are the key aspects of my development that I need to focus on? So as a part of that planning process, it's really, really important, and it seems an obvious thing to say, but it's very common that people come to us and haven't done much in the space of getting clinical input. So actually, there's a lot of focus often. You're very excited, you've got a potentially really transformative therapeutic in your hands, but actually the focus is often about, what's the cell doing, maybe how it's performed in an animal model. But actually what this is about is really, what is the physical product going to look like? And then how does that clinical procedure look? So, you're considering things like, how much do I have to make? Is it cryopreserved or fresh? Some of the logistical concerns. So is there a hold time? Do I have to do a special procedure in clinic that's going to require a specialist piece of equipment? Maybe there's some parallel development that has to go on there from a technology provider, for example. So it's really, really good to have this, again, as a living process, but to start to understand how that final product's going to look, so you've got something to work towards. So you've done a lot of planning. It can be kind of overwhelming at times. Where do you start? What should I do first? And generally speaking, it's a really good idea to do a risk assessment and a prioritization plan. So this is an exercise that can help you give you an idea of where to start. As I say, generally speaking, if it's going to be really, really difficult to do later on in your development, then it's probably better that you do it early on. But for most of the risks you can look at, you essentially go through your entire process. You're looking at the risk to the success of that process. What risks are going to cause problems? The likelihood of those risks then occurring, the degree of impact if those risks do occur, and then reflect on what that mitigation strategy should be to reduce the impact of those risks, and how easy is that mitigation strategy to implement. And if you focus on the degree of impact and the ease of implementation, you can kind of score each individual risks based on priority. So your high priority items are generally things that are easy to do, but are going to have a real big impact on your process. And as you move away from that, you're getting lower benefit and they're more difficult to do. So some of these lower priority things are often future-proofing activities, polishing, that kind of thing. So just a few examples here. As an example of a high priority would be perhaps with an allergenic iPSC product. So there's a lot of variability between pluripotent stem cell lines. So actually changing later in your process could be really, really potentially detrimental to the success of your development. So you really probably want to look at doing that and locking that down early. A medium priority example, so maybe the use of something like FBS. So contrary to popular belief, you can actually take serum forward for GMP use, but it's going to require a lot of QC testing to prove the safety of that product. And you also maybe have to consider securing a large lot of it that's going to be able to cover the entirety of your manufacturing. If that's suddenly unavailable for whatever reason, then that's going to potentially cause you a problem. So the risk is there, but it's not necessarily a complete full stop. And then on the low priority side, you might be looking at something like an alternative supplier. So this is sort of, a critical reagent that maybe you've already got 2 suppliers for. It would be handy to have an extra one, but, perhaps it's not something you need to worry about for the time being. So I'll just briefly cover a few of the common challenges. And I know James is going to talk to this particular one later on in the webinar, but generally scale is a huge factor, and it's usually the first one that people would cite when they're looking at the technical development of their product. So an existing process is often something like this, maybe tissue culture ware, often open, manual processes, generally fairly low throughput. You might be doing concentration and wash with something like a standard centrifuge. And essentially you're looking at 2 potential options of how to take your scale strategy forward. So on the scale out side, you're essentially kind of parallelizing your current process. So you're increasing the amount you can produce by having kind of multiple parallel streams. It's often really popular for autologous products for obvious reasons, but actually lower dose products as well. Sometimes things like the eye or the ear, for example, do take this approach. And again, it can often be a bit quicker. So it's, it's probably the least development from what you've already got. This, might get you there sooner. So if you've got a kind of a particular goal of getting first in human, then that might be an option for you. However, for certain therapies, that the level of scale that you're going to be able to get with this is just not going to be there. There's going to be variability across your parallel streams. The throughput is limited and the footprint is potentially very large. So then you're looking to kind of traditional scale-up bioprocessing type technology. So things like bioreactors, downstream technologies like I've got an example here, the KSEP and the LOVO, which are high throughput wash and concentration apparatus. And essentially they have a lower throughput. They're usually associated with lower cost, and I'll come on to speak about that as well in a minute. So cost of goods. So cost of goods is a really important thing to consider. It's not often the most important thing that people are looking at, particularly early on in their development, but it becomes much more important as you get closer and closer to clinic, particularly when you've got competing products in that space. You know, you have to make sure that your product's going to be able to be affordable and reimbursed by the various healthcare providers. So some of the things that would impact those cost of goods, reagents and consumables is an obvious one. Are there things that you can do, optimizations you can do to reduce the amount of consumables you need? Sometimes the answer is no, but it's good to consider that. And actually, you know, that can really help save costs during your development if you're financially limited as well. Equipment, another obvious one. There may be some kind of upfront costs that are going to save you costs in your manufacturing. Important to understand what the context of your equipment you're going to need. So if you're going to need to go to that large scale, for example, that investment is going to be important. Another consideration is that your contract manufacturer of choice may or may not have access to particular types of equipment. So it's good to liaise with them as you're doing your development if you think you're going to need that. Otherwise, you may be stuck with purchasing that piece of equipment for your manufacturer. License is another issue, often overlooked. We work in a complicated space. To be able to license various technologies, to be able to have the freedom to move forward and manufacture your product for commercial use is a real bottleneck for certain products. Labor is a major one for cell and gene therapy products. Generally, a lot of the processes that we use at the moment are very manual, generally require quite high skilled people, and there's obviously cost associated with that. And facilities, again, linked to this, clean room facilities are very scarce and very expensive for this kind of manufacturing at the moment. So, you know, how you design your process is going to really impact on the amount of space that you need. So another challenge, and I've already touched on it, is the open and manual processing that is really common with early stage therapies. And what we're trying to kind of move towards is more of a closed, automated setup. So the main issue around open processing is a sterility risk. And actually that can be very, very high, you know, depending on the type of process, the length of your process, you know, you're looking at potentially 20% batch failure. And that's just going to be really difficult for you to maintain your obligations to your healthcare provider, but also to keep your costs competitive. And it's often linked in with this manual processing, which is problematic for a number of reasons. It's, you know, you're more than welcome to take forward your manual process, particularly if it's going to get you to a particular developmental milestone that you need to demonstrate. However, it is going to be limited in its throughput. It's going to be inconsistent, particularly across multiple operators. It's going to be highly skilled. Difficult to transfer is a key one. So if you're trying to move between different manufacturing sites, or you need to standardize across multiple manufacturing sites, that's going to be impacted by manual processing. And ultimately, as we've touched on, it's expensive. So it's a major consideration if you can move to an automated system. And you can do that in something as simple as, you know, your cell counting, for example. The final challenge I just wanted to touch on is around analytical monitoring and process control. So what do we mean by that? So essentially, it's how do you know how things are going as the process progresses? Often cell and gene therapy processes can be quite complicated. They can be quite long, and you know, there's a lot of cost associated with that. So really, you want to know as soon as possible if something's going badly, so you can potentially fail early and fail cheaply, and then potentially start again, so you can, you know, get closer to meeting your timeline that you were hoping to achieve. And generally, this is the kind of journey your analytical development will go on. And I'll touch on this with some examples in a second. But essentially, you're moving from a kind of what happened approach to understanding why that happened with your process, what will happen, And then ultimately, how can we make it happen, this prescriptive? And then this is where you're kind of getting into linking with the automation for feedback control. So it's really important. It's really important to understand what features and quality attributes are linked to the efficacy and the quality of your product. And that's going to become more and more important as you move forward to phase 3, where you need a mechanism of action and ultimately a potency assay. So just as a few examples, just to sort of highlight what the cell and gene therapy Catapult doing about some of these challenges. The first example I've chosen is a closed automated scale-up of pluripotent stem cell expansion. So we've done this with traditional stirred tank reactors. So we take advantage of the fact that we can grow the pluripotent stem cells as aggregates, so they're self-aggregating aggregates in suspension. We do some parameter optimization at the 15 mil scale, scaled up to the 250 mil scale, and then ultimately a larger, more industrially relevant scale of the 1.5 litre scale. You can see from the graph that there's good consistent scaling using our parameters, and we maintain a very high viability, high cell density. We can ultimately get about 3 billion cells from this process in a 1.5 litre STR. The pluripotency and the quality of the pluripotent stem cells is maintained. They can differentiate into all 3 germ layers. And basically, you're getting a serious reduction in the amount of labor required to operate this process. A huge 180% increase in the yield, in the productivity, and a significant 33% reduction in the overall cost of goods. So over to the analytical monitoring side, I've got an example for monitoring AAV production, and this is using Raman spectroscopy for real-time monitoring of what's happening in your system. And it basically takes advantage of the fact that Raman spectra can be used to fingerprint various analytes and various molecules in your spent medium. And what we've been able to do, as demonstrated by the data here, is we've basically been able to identify and monitor various metabolites, traditional metabolites within the media, so glucose, lactate, etc. And these correlate very well with the crosses, which are our offline orthogonal measurements. So we're getting that real-time monitoring of cells. We've also developed a surrogate model to understand the cell growth. So we know how the cell growth is progressing using the same system. And here is an example of how we've taken that one step further to understand how we can monitor in real time the vector genome model, so basically the AAV production. So essentially, we've taken the same approach and we've managed to find a surrogate model that tracks pretty well with the AAV production compared to our orthogonal offline measurements. And the idea is here, not only do we have that kind of process performance readout, we're also moving towards that process control, that feedback model that I talked about earlier on. So essentially, in one system, you've got continuous online, anywhere, accessible monitoring of both, the metabolomic information, the amount of vector you're producing, and the cell growth as well. So you can see how this is going to really help to optimize things like feeding strategies and ultimately the production itself. So thanks everyone for listening. I'm happy to take any questions. If you think there's anything I or the Catapult can do for you, please don't hesitate to get in touch. And I'll leave it there. Thank you very much.
Thank you for a great presentation, Matt. That was really interesting. So we'll actually take the questions. These are Matt's details if you want to get in touch directly. But we'll leave all the questions then towards the end. But if now, I'd quite like to do the poll now, because we would actually just like to know a bit about those that are participating. So we've got some questions. I'll just launch the poll now. Right, so I'll give you about 45 seconds, I think, so to answer the questions. So question 1, what stage of process product development are you at? 2, do you have an upscaling strategy outlined? 3, what is the current priority with regard to developing your process? And what do you envisage your major manufacturing bottleneck will be? Number 4. And 5, how do you count your cells? Quite interesting. So I'll give you just maybe another 10 more seconds because they're quite long. You have to scroll down to get to all the questions. And then I'll share the results before we start the Q&A. But I can see actually quite a few people are still answering, so I'll just leave it a bit longer. Let's see. I think, well, you're still answering. Sorry, maybe the questions were too long. It's taking a while for everyone to keep answering. Okay. I think it's pretty much finished now, right? I think pretty much finished answering. Right, thank you very much. I will end the poll and then show you the results at the end. Okay, end polling. And now if we could hand over to James. James, please.
Hi, Anna-Mika. So thank you very much for the presentation, Matt, and thank you guys for inviting me to speak. So I'm going to be talking about some of the key challenges that are faced in the process development to manufacturing roadmap, and focusing on the scalability challenge and the translational work that's needed to mitigate against potential pitfalls down the line, and some of the potential tools and strategies that we can use in order to help us along. So why is translation work important? Well, cell and gene therapies and advanced therapies have processes that are extremely expensive, time-consuming, and highly complex. As a result, they've got extremely prohibitive costs associated with them. There's a lot of batch failure, as Matt alluded to. This can be due to contamination, manual error, and quite a lot of the times due to low process control, so not understanding your process. And also the long lead times that are associated with these therapies, again, add to that time and resource intensive utilization. So it is therefore important for us to have robust translational work in place to help mitigate against some of these issues. So we like to think that good translational work has the motto of start as you mean to go on. So from the beginning with your protocol generation, think about what you envisage your final process to be, because each part of your process essentially leads to the other part. So here we need to be starting to think at every point of our product development timeline, from process development all the way to manufacturing. Is it sustainable? Is it scalable? And also, what are the technologies that I need in order to help me progress with my journey? What are the key kind of critical quality attributes of my process? And also starting to understand from the get-go, you know, what are the reagents that I need? Can I actually use them? Are they scalable and sustainable? And also understanding what are the cell characterization techniques that I'm going to have, or process characterization techniques that I'm going to have. What that allows us to then do is go on to kind of the protocol adaptation stages. And at this point, we're looking at optimizing those protocols, standardizing those protocols, and also using that prior knowledge from the cell characterization to start to develop our assays. So this is really important. So the more data we have, the more powerful it is. So here we can start to develop assays, not just for quality, but also potency, identity, and even safety, for instance. And ultimately, we want all of that work package and all of that translational work to allow us to develop to a GMP manufacturing standard, where we can have that robust quality control, but also importantly, we can manufacture the clinically relevant and commercially relevant cell numbers. And also we're starting to have a strategy of how we're going to sort out our logistics and our storage, and also ultimately what that's going to look like in the clinic to the patient and also to the payer. So it's really important, as I have alluded to and that Matt has alluded to start as early as possible. So really think about what the future of your product is going to look like. Try to see how you can make decisions now that will put you in a better position. So this kind of roadmap is kind of showing that, you know, from the key conception stages, we need to be thinking about defining that mechanism of action as much as possible and seeing how that relates when we're looking at the preclinical studies, understanding about the functionality, and then also when it comes to that process transfer, this is is where we need to be nailing down what are going to be our key critical quality attributes, what are key product quality target profile, what is also their process parameters, and understanding as much as we can in terms of risk, so carrying out those activities that Matt alluded to before. And then that can then allow us to then translate that process once we have that understanding of what we're trying to achieve, what are the key things we need to measure. Therefore, we can have a process that's easy for us to characterize and also easy for us to transfer, and also something that we can hopefully be able to validate in order for us to get to that manufacturing stage. So this kind of Ishikawa diagram or cause and effect diagram is just a good tool that we like to use and we encourage people to use when it comes to kind of trying to understand your process. So again, think about beginning with the end in mind. So take the time to analyze your process. And this can be done preemptively and proactively, or by looking at historical data you might have. So think, okay, if my process is going to fail, what's going to be the cause of that? What are the different categories? Is it the people? Do I need to improve my training? Is it my method? Is it not standardized enough? Is it the environment? Is it the equipment? Do we need to have better calibration? Or is it even my materials? Is something out of date? Do we have batch variation? And all that kind of thing is really important to be thinking about. And if we can go as granular as possible at this stage, it's really beneficial, because that can allow us to have that kind of context in order for us to move forward and know where we think the potential pitfalls might be. So here it's really important to be thinking about the best sort of technologies and procedures that are important for your process, because every single process is going to be different. And it's important that you do the work to investigate and find the best things that are going to work for your process, and start to think about how technology can aid with the process. And this can be aiding in terms of the quality, in terms of the scale, and also in terms of just capability as well. And also thinking, how do I monitor the process? Like Matt was saying, it's really important to know what's going on within your process. So this is where process analytical technologies really come in handy. So being able to monitor and understand your process as early as possible will help you in the long run, because it can start to get that really granular detail, but also you start to understand what's my signal against what's my noise. And the earlier you can start to understand that, the better. And also from a kind of market authorization and regulatory point of view, the better you can kind of provide rebuttals for anything that might come up, the better it is for you. So it's also important to think about looking into what are my scale-up technologies, what are my end-to-end solutions, how can I automate things, and what are those kind of key process analytical technologies that I can use. So in terms of the scalability challenge, there's a plethora of things that need to be considered. So at Microfluidics, we are working on technologies that aim to address some of these challenges, mainly from a process control and reproducibility angle, and also from a cost perspective. So by working at a microfluidic scale, what we like to harness and kind of use to advantages the need for that lower reagent usage, but also having that more precise control of the cell environment and the cell milieu, also having very high yields and being able to be amenable to automation through the use of pumps and valves, for instance. So here's just a little bit of a case study of some work that we've done with our technology, showing the advantages of that we can harness from being in the microfluidic scale. So on our device, we can essentially do end-to-end bioprocessing. So on our device and on a single chip, we can do the seeding, we can do the transduction, we can do the expansion, and also the harvesting. So this can be amenable to almost any sort of process that you can think of doing in terms of cell manipulation. So the transduction could be swapped in for cell differentiation or any sort of other kind of manipulations. And this is something that we can do in a closed manner by automating the processes, and we have extremely high efficiencies, certainly in comparison to some of the work that we've been doing. And ultimately, this can be something that can be integrated with technology, with analytics, such as your key sensors for oxygen, CO2, pH, temperature, etc., and even more analytics as well. And this is just a little bit more kind of data showing that we can have really high efficiencies in terms of our harvesting. So here we start off with cells that you can see here, and you can see both in this bright field and the GFP that is extremely high efficiencies when it comes to kind of harvesting the cells. And this is really crucial with some therapies where every single cell counts and we can't afford to go back to the patient to try and get more materials. And we can do this whilst achieving really high cell densities without compromising the cells. Here's just some images of some counts that we've done. We've got over 94% viability, and this was done on the NC202. But how do we actually scale up? So from our perspective, what we aim to do is kind of grow with the process and the different therapies that are out there. So we can start from that really early stage with that discovery stage where we're doing kind of the proof of concept work, starting to kind of understand what I think I need my cells to do. And then we can then progress on to going to a more process development stage where we can essentially have more chips and cassettes. And here we can start to add analytics to start to add to that powerful data to aid in that process development. Once we have a better understanding of that, we can then progress on to the more clinical or commercially relevant manufacturing scale, where we can lock down our system, but still have the ability to monitor what's going on. And this is the ScaleUp strategy that we aim to use in order for us to help in terms of process control and also the cost challenges that are faced within the industry. So here, I'm just showing some of the ways that we're looking to address the scalability challenges. So we've mapped out the different use cases of our technologies, and we can see that we can be part of the whole product development pipeline for different users. So we aim to offer solutions that essentially can seamlessly scale up with your product or your process, whilst removing the need for platform and environmental adaptation studies. So essentially what this allows us to do is save you both time and financial resources, because due to the way that we're scaling up, the microenvironment that the cells are exposed to from the discovery stages all the way to the manufacturing stages is essentially the same. But most importantly, that environment is something that you can have control over and can scale to suit your needs and your processes. So we've been in contact and partnering with a range of different partners throughout this kind of whole product development scale. And we have some exciting projects that we've got coming up. And I would like to end there and say I'll take questions in the Q&A. And if you'd like to hear any more information about us or kind of talk about any sort of collaborations, then please feel free to get in touch.
Thank you very much, James. That was a really interesting presentation. Thank you very much. So I promised to share the results of the poll. So let's have a look. See if I can make it share results. Let's see. So what stage of process product development are you at? So the biggest majority, feasibility studies and preclinical studies. So already interesting. Do you have an upscaling strategy outlined? Yes, started creating a plan. So that's 33%. Yeah, and 14% yes, plan is set and resources allocated. Okay, what is the current priority with regard to developing your process? First in human clinical data. Oh, interesting. Okay, what do you envisage your major manufacturing bottleneck will be? Scale. The vast majority is scale. So I think your presentations, both of you, are spot on. And how do you count your cells? Nuclear counter, other cell counter, and hemostator. Oh, that's interesting. Is it quite an even spread? Okay. Right. Well, thank you very much for sharing that information with us. It's really interesting to see what everyone is, stage everyone is at. So stop share results. Okay. Right. So questions. So we've got some questions in the Q&A section, but we also had some questions emailed in. So I think this one might be for you, Matt. Do you think integrated or modular bioprocessing is better for autologous products?
Thanks, Anamika. It's an interesting one, really, because I think, again, it comes back to that conversation around your priorities are. And I think the integrated systems, particularly for the autologous immuno-oncology space, have been really successful, I think, in allowing some companies to kind of get their manufacturing to a level where they can get through their clinical trials and ultimately get to the market. I think in the conversations we're having with collaborators now, it seems that actually that next step of development, that kind of industrialization, industrialized manufacturing step, may actually revert back to something more modular, because of the fact that the integrated systems, once you've started a process on there, it's kind of out of action for a matter of time, and all the various individual components of that. So things like the electroporation, the growth, the concentration and wash are all occupied, whereas actually the modular systems have the advantage of, you can kind of stagger processing for more patients. So I think we're probably seeing a shift back to that now the products are reaching the market.
Right, okay. Okay, then the other question we got was, what are the key questions to ask technology developers, companies, when working or planning to work with them as a therapy developer? James?
I can take that one. Yeah. So I think firstly, as iterated in the presentation, I think it's really important to know your process, because once you understand that, you can start to have targeted questions. So, you want to be asking or looking into how mature the developer is, or the specific technology that you're thinking of. So why is that important to ask? Well, because it can give you an indication of their flexibility, which might be something you want. So you may want something that's fairly flexible, or you may want something that's locked down. So both options have kind of pros and cons. But also I think an important thing to look into is what is their quality management system like? Has it been done in a way that will make your life easier, or will you actually have to spend a lot of time validating their work because it's not, of the right kind of standard? And that can really have a big impact in terms of financial and time resources. So I would say a good company will have the right ISO standards and GMP kind of modes of working, and they will be more than happy to share that information with you as part of kind of like your standard auditing before working with key suppliers.
Yeah, okay, got you. Okay, thanks for that. Then we've got also questions in the Q&A. So what are your methods to standardize your protocols for the GMP protocol optimization?
I can take that one if you like.
Yeah.
Great. So yeah, I think again, it comes back to that conversation we were having about really understanding your process as early as possible. So I think it's about understanding the critical processing parameters that impact the success, however you define that, of your product and your process itself. So I think once you can understand what the key aspects of your process are, then you can add in that process control, whether that's the need for specialized monitoring at that stage, or a different piece of equipment that can kind of give you more consistency. That's going to really help you in terms of your standardization. But it really hinges on having that understanding in the first place, and then you can start to tighten that process up, set thresholds, acceptance criteria for various stages before moving on to the next step, for example. So that, as I say, I think just to reiterate, it's about having that understanding, and then really starting to nail down the detail in how you perform things. Okay.
Yeah. Right. So next question we have is PAT methods used for process monitoring. How much validation is necessary? Do you only use those for monitoring or also for process control, e.g. automated feedback regulation of the process?
So I can take that one. Yeah. So I think with the PAT methods, it's about validating it for your needs. So I think you can have process analytical technologies that can give you a lot of information, but you need to have things that are valid and robust enough to kind of give you the information that you need. And then once you kind of have the idea of what that mechanism of action and what you're trying to achieve is, you can get the right sort of tools that can give that. In terms of the automated feedback, that is something that's really interesting. And I think there's a lot of benefit to that, but that requires a lot of validation in order to kind of fully ascertain that, you know, if I have X surrogate marker and I see X, Y, and Z happening, that's definitely going to lead to either my batch failing or, you know, proceeding how I expect it to. It's something that I think a lot of people really like the idea of because it has so many advantages, but I think, yeah, it still needs a little bit more work.
Yeah, I agree. I don't think we're quite there yet on the feedback control, but I think what's important is that the technologies are catching up with the to be able to support that in the future. So, you know, the biomarker discovery, etc., and the process analytical technologies hopefully then are in place to then start to build those models and get that process control.
Okay, thank you. Let's see, we'll go to what if you don't quite know the mechanism of action? How does that affect analytical development and scale up?
I can go with that one if you like.
Yeah,
I'll go for James.
Go first.
Oh, thanks.
Sorry. Okay, we'll go for. I think you took the last question, didn't you, James? So we'll go to Matt. Yeah.
Great. Okay. Thanks. Sorry, James. So essentially, I think. Sorry, which question are we on? The mechanism of action question. So essentially, you don't necessarily need the mechanism of action until the phase 3 trials. However, it's really valuable. And the reason you don't need a mechanism of action is you essentially don't need a potency assay, which is basically an efficacy assay, until that phase 3 trial. Why it's helpful in terms of your process development and your scale up is that it comes back to what we've touched on previously, which is if you understand what's important for the efficacy of your product, then you can link a lot of your process control to the endpoint, to that mechanism. And actually, that's really beneficial for your process development because it gives you something to aim at, and it means that you can screen against that kind of product quality. So it's not essential in that early stage, but if you can start to think about what that would be, it's only going to be beneficial to you later on.
Yeah, I would agree with that, and I think it really just helps you to kind of pick the right sort of process stage gates as well, and also the technologies you need to go with your process as it develops. Kind of as I was alluding to, everything kind of feeds onto each other. So if you can think, actually, at my stage 3, I'm going to be needing this, you can start earlier on to have the right things in place to allow you to show that with a bit more ease.
Yeah.
Absolutely.
So I guess this one really is for James, because is the microfluidics applicable for suspension cells?
Yes, so the technology is for both suspension cells and adherent cells. So we have work streams that focus on both. Yeah. Right.
Okay. And then what are the key strategies used to achieve a good quality product? Is that Matt's turn, is it?
Yeah. Okay.
Or is it James' turn? I can't remember. No, it was Matt's turn. That's right.
Thanks, Anamika. So as I've covered in the kind of presentation there, I think it's really around, you get that understanding, and then I think one of the things actually that I didn't touch on, but it was in the presentation, is good data management is really important. So actually, particularly even at that early stage, particularly for academics, it can be often difficult to keep that data well recorded and in a kind of usable fashion. That can be really helpful later on as you need to refer back to various things, particularly as you start to make changes to your process. If you have a kind of broad understanding of, if I change X, I've got a window of concentrations that I can use, and it's not going to have a huge impact on my downstream product, for example. That's really beneficial because it starts to give you that flexibility in what you can get away with changing as you change equipment and things like that. I think it really depends on what the context of your goals are, what the context of your particular type of product is, but basically it's around that planning and understanding what it is you want at the end, and actually that has a lot of impact on how you then get there.
Yeah. Okay, so now I think we've got another one, what are the different processes involved in the gene therapy manufacturing which are not involved in other dosage forms? James?
I think Matt can take that one. I think they have more experience with
Yeah, so if I understand the question correctly, I'm assuming you're looking for the differences between sort of vector manufacture and perhaps cell manufacture. So I would say there are two kind of main areas that are quite different. The first being the analytical monitoring of those processes. There's a lot of kind of specific tests, particularly release testing, etc., that you would want to check your vector for. So things like empty capsids versus full capsids, the infectivity, the efficacy of those vectors, etc., that you may not see with other products. I think the second, and I should add, there's a lot of effort and development going on into those sort of analytics at the moment, not least by the Catapult, who have a platform here as well. But as I say, on the second hand, I think it's around that downstream processing question. So there's a lot of kind of cleanup and clarification that needs to be done of those gene therapy vectors, which you don't really see in the cell therapy space. So kind of methods of kind of chromatographic methods, et cetera, that have kind of transferred over from sort of traditional bioprocessing for things like biologics and monoclonal antibodies. So I would say that those are probably the key big differences.
Right. Okay. We've got another questions just coming. Is there a lot of DOE involved in cell and gene therapy process development?
Yeah, I can take that one. Yes, there is, and I think it's one of a key tool that is important to use. So kind of like what Matt was alluding to before, it allows you to get an understanding of what you'd call the design space. So understanding what your critical process parameters are can then allow you to understand what your critical quality attributes are. And this is all stuff you do in that kind of process development stage, and DOE and quality by design are some of the key tools that can be used in order to kind of get that very robust information, because a lot of DOE is really based on getting the best and most well-formulated data and ways of carrying out your experiments in a way that will save you time but give you the most kind of value for your efforts. So yeah, it's an important tool.
Okay, so then I think we'll come on to our final question before we end this webinar. So what is the process of converting a product from clinical grade to GMP grade?
So I can take that one. I think if I understand correctly, again, a clinical grade reagent as such is something that's already approved for use in patients, whereas a GMP, the good manufacturing practice, is essentially a quality management system that allows you to see the traceability, the safety, purity, and ultimately efficacy of your product that you're going to hopefully then be approved to kind of take forward for clinical use. So really, it's probably the other way around. It's actually, how does GMP support clinical access, essentially? So that's a kind of a set of values or sort of mechanisms which allows you to kind of find that traceability, etc. And ultimately then, you'll be hopefully rubber stamped for clinical approval. So it's probably more the other way around rather than going from clinical to GMP.
Right. Okay, well, thank you. That brings us to the end of the webinar. There was a question about will we be able to share the presentation, so that this webinar will go up on our website and you'll be able to access it from there, probably in a few days. But thank you everyone for joining us today, and have a lovely rest of the day. Bye.
Thanks everyone.
Thank you.
Bye-bye.